You're doing signals on what?
Electroporation.
Ah, really?
Beautiful, excellent. So glad.
We're trying.
Wow. And what's in the pipette? It's in the electrode.
Okay.
Channel. Oh, okay.
In CA3 pyramids.
You make a new file there?
So it's going to go back in the incubator after you...
Yeah.
And so this is
Hopefully it's not contaminated.
so it's on. It's DNA. It's not.
cDNA for channel adoption, and then a protein that should alter our synaptic function.
So we want to also a single cell electroporate SLM1, right? There is a RNA binding protein that should, be expressed presynaptically, but we want to see if it does something in the postsynapse by patching the neurons that are surrounding this.
Wait, this is a. You have a RNA that you're trying to follow.
No,
Is that right?
it's an RNA binding protein
Right.
that regulates splicing of the cell tissue molecule neurexin.
Okay, so there's, I know that there's this way you can tag RNAs that are being translocated.
No.
Not the MS2 system, this is really an RNA. So what's the RNA binding protein doing? What's the...
This is
We
the
don't know the
experiment.
function yet. That's the cool thing.
Aha!
But we know a few things, eh?
Yeah, we know a few things.
But is it...
Regulating splicing of multiple
right?
factors.
Does it leave the nucleus?
No.
Okay.
It stays in the nucleus.
But you know that it regulates synapses in some way?
Yeah, by splicing of a cell tissue molecule.
Isn't that SLM2 already in these cells? Why are you adding it?
It's a misexpression actually.
That's a mis-
The cool thing about these proteins is that SLM1, which we are misexpressing, is normally only in the dentate gyrus.
Uh-huh.
And SLM2, homologue, is in CA1, CA3. And now we are misexpressing SLM1 in these cells, trying to figure out whether this changes presynaptic modulation of neurexins and therefore also the postsynaptic.
Ah! This is one of the neurexin regulating the alternative exons.
Yes.
So that's why you care about it.
That was a test.
Yeah. Okay.
Yeah, it regulates. So it's like what Joris was saying today, that you can get a plus or a minus isoform.
Yep.
X4, which means that either exon 20 is included or excluded.
Yeah.
And this changes the binding affinity of neurexins
This
to
is-
neuroligands or LRTM.
This is in the beta version as well as the alpha, but it's in the it is in the beta. But at least we understand the beta version
A
better
bit better, yeah.
than the alpha version. And so this will be the CA3, CA1 version.
Yeah, so we are going to, so we put channel rhodopsin in there as well, so we're going to
Yep.
activate the cell body and then record from a non-infected cell.
Yeah.
So we're going to check CA3 connections.
So is this, no. Is this intron? Does this interact with? It's known to interact with which proteins?
So
The differential splicing interacts with which
yeah,
proteins?
so the plus isoform
Yeah.
usually interacts with CBLNs, for example, which are CBLNs, for example. The minus isoform interacts with LRTM2
Okay.
and neuroligin 1, 2, and 3.
And this is the minus version?
So
or
the RNA binding protein normally regulates the skipping. So we are
Yep.
forcing the cell to produce more minus isoform.
Yep.
Therefore, it should recruit more neuroligins and LRTMs, and therefore it should also recruit and stabilize more NMDA and AMPA receptors. And that's why we are going to measure NMDA and AMPA receptor ratios in our experiment, but also we are going to check for peri-pulse facilitation, to check for presynaptic effects, because we don't know yet.
So you think that it will change the strength of the synapses, but not the specificity?
Yeah, it could be.
Or it could